absorbent assay. So what we do here is that we look at various concentrations of the sample to see the reaction. You start with, and you can have these little plastic trays with 96 wells in them. In this well, you've coated it with, it could be the antibody or it could be the antigen. It could go either way. But let's make the assumption now that you've put the antigen in these wells. Okay? You expose the antigen to the antibody here. Now, what we don't know is if this is the antigen for the antibody that we're testing. So if we do expose it, if it is the right one, the antibody will bind to it. So we get our antibody binding to the antigen. Wash this away, and then we expose the wells again to a different antibody. An antibody created by an animal like a mouse that responds to this antibody. And so it will bind to it like so. But we've also attached an enzyme to this second antibody. And this enzyme will serve to cleave a colorless molecule. We'll expose this to this colorless molecule, and if the enzyme had stayed attached and is present, it will cleave that molecule and you'll get a color change develop. If that color change develops, then you know the antigen is present. And you can go the reverse direction, or I should say the other way, where you start with the antibodies. So you have your wells now treated with the antibody, and you expose the antigen. And so if the antibodies are there, the antigen will bind, and then essentially the same thing. A couple of videos I want to show you.
Monoclonal antibodies.
Got a picture yet? Okay.
Just press the button.
Monoclonal antibody preparations contain only one type of antibody, derived from a single cloned B cell. Consequently, they are highly specific for a single epitope and have applications, for example, in diagnostic microbiology and cancer therapy. The first step in producing a monoclonal antibody is to inject an animal with an antigen containing the specific epitope of interest. Each B cell produces a single type of antibody. B cells are isolated from the spleen and then mixed with myeloma cells, a type of cancer cell that grows continuously. Addition of polyethylene glycol causes the two types of cells to fuse together to form cells called hybridomas. The mixture of B cells, myeloma cells, and hybridomas are cultured under conditions which permit growth of only the hybridoma cells. Each hybridoma cell will produce a single type of antibody against a single epitope. The single hybridoma cells are then separated into individual wells of a microtiter plate and tested for ability to produce the desired antibody. The hybridoma cells producing the desired monoclonal antibody are then cultured. Monoclonal antibodies are isolated and purified.
That's it. Just kind of stops.
The enzyme link convenient sorted assay, or the enzyme link convenient sorted assay, or ELISA, is a commonly used format for serologic testing. The purpose of this animation is to explain how this test works. ELISA serologies are usually done in multi-well microtiter plates so that dilutions of serum can be easily prepared and tested. To better understand how this assay is done, let's take a closer look at what happens in one of the wells of this assay plate. To perform the assay, the wells of the plate are coated with the antigen of interest. For commercial tests, this would be done by the manufacturer of the assay. To begin testing, the wells are filled with dilutions of the patient's serum. If antibodies against the antigen are present, they will bind to the antigen fixed to the bottom of the wells. But only antigen-specific antibodies will bind to the wells. The wells are then washed out to remove all unbound antibodies. Next, a solution of an animal antibody against human antibodies is added. This second antibody is covalently conjugated to an enzyme. The wells are washed again, this time to remove any unbound enzyme-conjugated antibody. Finally, a solution of a colorogenic enzyme substrate is added. The interaction of the substrate with the enzyme on the second antibody generates visible color. The development of color in the wells with a specific antibody can be seen with the naked eye or quantified with an electronic plate reader. The reaction is less intense as the serum is diluted and the amount of antibody captured in the wells decreases. The titer is the highest dilution with definite color development.
Any questions, any comments? Well, I don't have any more notes, so I guess that means I'm done for this section here. So review on Monday, exam on Wednesday, Scantron 882E, bring a pencil. This is the toughest exam of the three so far. So just be forewarned. A lot of moving parts in this section here, okay?